Hello, everyone. My name is Diana Pashkova. Welcome to another episode of Canada's Secret Science Society. Today, we will be discussing a very important topic antibiotics. They relate to the origins of life unit as they work by targeting bacteria's biochemistry. Antibiotics, when translated from Latin, means against life. Antibiotics are a type of medicine that are used to kill bacteria in your body. They are used to treat infections but are ineffective against viruses. We might think that antibiotics were introduced only in the 1900s by Paul Ehrlich, but that is not the case. In ancient times, before people knew that infections were caused by different types of bacteria, their treatment consisted of different types of molds and plant extracts. For example, in Egypt, people used moldy bread to heal infected wounds. Mayan nations used to use mold to treat wounds and diseases of the intestines, and in the Middle Ages, Ibn Sina used mold in purulent wounds. There are many different types of antibiotics, and they can be classified based on their origin as well as other indicators. The origins can be molds, radiant mushrooms, bacteria, and plants. But today, we will be focusing on only two classes, the penicillins, which originate from molds, and the streptomycins, which originate from radiant mushrooms. In 1928, a new and life-changing discovery was made by Alexander Fleming, who was a physician-scientist from Scotland. He had noticed that one of his Petri dishes containing Staphylococcus aureus bacteria looked different from the rest. He realized that Staphylococcus aureus bacteria had been killed by the mold that was growing on the same plate, and had figured out that it can be used to kill many types of harmful bacteria. He then isolated penicillin from the extract that came from the mold. Penicillin later became the world's first mass-produced antibiotic, and its introduction began the golden era of antibiotics. Penicillin V and G are the only penicillins that are natural, the rest are semi-synthetic. Penicillin is white in color, crystalline solid that has no distinct smell. How does this work, you might ask? Well, let me explain. Penicillin kills bacteria by interfering with the ability to synthesize the cell wall. The bacteria increases in length, but is unable to divide, and as a result, the cell wall weakens and ruptures. In simpler terms, penicillin acts on the peptidoglycans, which is a substance forming the cell walls of many bacteria. It consists of glycosaminoglycan chains that are interconnected with short peptides. Peptidoglycans create a structure shaped like a mesh around the plasma membrane of the bacterial cell, which increases the strength of the cell wall and prevent external fluids and particles from entering the cell. When a bacteria multiplies, tiny openings open up in its cell wall as the cell divides. New peptidoglycans are then able to fill up these openings to rebuild the cell wall. Penicillin blocks the protein struts that connect the peptidoglycans together. This prevents the bacteria from closing the holes in its cell walls. As the water concentration of the surrounding fluid is higher than inside the bacteria, water rushes through the holes into the cell and the bacteria bursts. Now let's talk about streptomycin. Streptomycin is an antibiotic synthesized by the soil organism Streptomyces ghorasius. Streptomycin was discovered by American biochemists Selman Waksman, Albert Schultz, and Elizabeth Bugey in 1943. It was the first antimicrobial agent created following the penicillin, as well as the very first antibiotic that was capable of treating tuberculosis. Streptomycin works by interrupting the function of the bacterial cell's ribosomes. Ribosomes are the protein builders of the cell. They do this by linking the amino acids of the cell. Anyways, ribosomes have two subunits. The bigger builds the proteins and is guided by the type of RNA called messenger RNA, which connects to it. The little subunit's function is to read the mRNA and to choose the matching transfer RNA molecule that chooses and delivers the next amino acid to the ribosome. Streptomycin bonds to the small subunit, and as a result, it causes the small subunit to misread the sequence. This results in the synthesis of random proteins, which leads to killing the bacteria. In conclusion, antibiotics are a great drug that has helped many people survive their sickness, which at times could have been deadly. There are many different types of antibiotics, and because of their variation, the number of deaths due to infections has decreased drastically. If we continue to progress our knowledge about antibiotics, we could potentially save lives of millions. Thank you for your time. Have a good day.